biological sensing and olfaction. We have Peter Mombaerts here. So Peter is director of the Department of Molecular Neurogenetics at the McLaren Institute of Biophysics in Frankfurt. And Peter has an outstanding track record with studies of the olfactory system with many of vertebrates, mammals, with also a very strong developmental component. And why that is that the olfactory system has some very unique features that force it, if you will, to regenerate neurons and regenerate processes and so on, which means a very complex sorting and orchestration has to occur in the system to make it all work reliably. And Peter is really at the forefront of studying and understanding this system, so it's really great that Peter joins us here today to explain everything about the olfactory system of mammals. Thank you, Peter.
Okay. Thanks, Paul, for inviting me. And thanks for staying. I will go a bit into your lunchtime. There is a resonance, but not too much, I hope. I have my first slide.
We have to switch.
We did that already.
No, I don't think so.
Well, we're testing it here during the pause.
Ah, okay.
Yeah.
They forgot.
But he somehow likes this slide. So. Let's go back to that one. I think mine is cooler, but
yeah.
Look at that with the little movie.
That's why you went to bed so late,
Yeah.
huh?
I just changed my affiliation, by the way. That's my new title, Max Planck Research Unit for Neurogenetics. Okay, so I have to give you a bit of introduction here about the molecular biology, genetics, and anatomy of the olfactory system, because you probably haven't heard so often about it. And this field of olfaction really has a before and after 1991 stage. Before 1991 perhaps was not as interesting, but in 1991 a paper got published in Cell by Linda Buck and Richard Axel, and for that paper they got the Nobel Prize in Physiology of Medicine in 2004. And Buck and Axel, Buck was at that time a postdoc actually for Richard Axel, so it's a bit unusual to get a Nobel Prize for your postdoc work. They come up with genes that encode G protein coupled receptors, and it was known from work in the 80s by chemical work that the pathway was a G protein pathway, a traditional signal amplification. Very large number of genes. That was the most stunning finding in this paper. In fact, the initial discovery was in rat, not in mouse. And now that the genomes are sequenced, we know in the mouse there are no less than 1,200 intact genes plus a few hundred pseudogenes. In the rat, even more, 1,500, and there are some rumors about rabbit and elephants with 3,000 odor receptor genes. I mean, absolutely crazy. This is much more than any other gene family in the genome.
One question, Peter, from that. If you see the number of genes encoding these receptors expand, is also the total genome expanded?
No.
No, so it really takes over territory in a sort of a fixed size genome.
They're very small genes without introns in the coding region, which is very unusual. So they can amplify very easily. They don't take too much space, we can say it this way. And several people, this is of course a fantastic subject for evolutionary studies to look at positive and selection and so on, and that's been really hard. There's a big review written a few years ago, I think the best one, by someone comparing what we then knew about the odor receptor genomes in different species, and he says it's basically random. He couldn't see any evidence of positive selection. Now, how can you put positive selection, Darwinian selection, on a family of 1,200 genes, right? A mouse that loses one gene in evolution or gets an extra one, or another species, will it be more fit than the other ones? It's a bit hard to think about that. So yes, that's the current thinking. In that paper, so these are the 3 lines of evidence by which Buck and Axel put forward an hypothesis, which really was an hypothesis then. We have cloned the odor receptors that are also expressed in the nose. So if you have a very large number of genes and an extremely large family of genes that encode receptors of the expected type, 7 transmembrane proteins, G protein coupled receptors, and they're only expressed in the nose, then of course the best bet is that these are olfactory receptors. It took, however, many, many years before the first ligand was found. For a pharmacologist or biochemist, a receptor is only a receptor when you have something that binds to it and either stimulates or blocks it. And it lasted until 1998 when Stuart Firestein at Columbia University, independently from Richard Axel, who was also at Columbia University, found the first ligand for a receptor. And still today, 2013, is still one of the biggest problems in our field. For very few receptors do we have ligands, so most of these receptors are still orphan receptors, as some people say. And conversely, for very few ligands of particular interest, do we have receptors. That is one of the top problems to be solved in the field, finding an easy way to go to receptor-ligand relationships. And I won't discuss that today. But closer to what I'm interested in, also what I will talk about today, half the time, is the gene regulation problem. It was apparent from the first paper that not all these genes are expressed in all neurons. That wouldn't make any sense, right? You couldn't do any coding if all these 1,200 genes are co-expressed in the same cells. Very quickly, the literature came upon this notion that there is one gene per neuron. The question is, how do you prove that? It's a good topic maybe to discuss once in a student seminar, because it's very hard to prove that, right? That of each mature neuron, only one gene of these 1,200 is expressed. In fact, I summarized that in a somewhat critical review about 10 years ago, Current Opinion in something, neurobiology, I think. And now that paper gets always quoted for one gene per neuron, but basically we don't know. Okay, I would not be surprised if there are exceptions to that. There are specific pairs of receptors co-expressed or random pairs that's all still open. We have to take it with a grain of salt because this statement is very difficult to prove conclusively and also actually very difficult to reject because every time you have evidence for two genes co-expressed, people come up with excuses basically. Okay, so it's a bit of a conspiracy here. If we all keep saying there is one gene expressed per neuron, that makes our life much easier. Now, lymphocytes, B and T cells, also have a similar case. Many genes, many immunoglobulin genes, one antibody per B cell, one T cell receptor per T cell. I did my PhD on that. And there it was much easier to establish that because you have cell lines. You can take one cell, make lots and lots of them, or there are naturally occurring tumors, leukemias, lymphomas, what have you. And then you have a lot of material to show that. In our case, we can only look at primary neurons. We cannot grow these cells, we cannot make cell lines. That's a generic problem in neurobiology, that you cannot make cell lines of mature neurons. Okay, so I've just qualified that, but for the rest of my talk, I will just not criticize this dogma anymore, that there is one gene expressed per neuron. But what is well established by a few techniques and by several labs is a very peculiar phenomenon, that of that gene only one allele is expressed. So most genes, you may not think about it like that, but most genes are expressed in two copies, right? One copy comes from the mother, the maternal allele, the other one from the father, the paternal allele, and they're co-expressed. So the default mode of gene expression in mammalian systems, I think in most vertebrate systems, is biallelic, about 50% of each. There are some genes that are an exception, of course, genes on the X chromosome in females, because one X chromosome is inactivated. Immunoglobulin T cell receptor genes are also expressed from one allele. Then you have all the imprinter genes, where the expression is dependent on the sex of the parent. So if it comes from the mother, it's expressed or it's not expressed, or the other way around. But this is yet another example of that. In a given mouse, be it male or female, of the cells that express given receptor gene A, half will express it from the maternal allele and the other half from the paternal allele. And I will show you a very nice visual demonstration of that. Very unusual property, not understood why, let alone what mechanisms. And it could be that these two features there, one gene per neuron monogenic and one allele per neuron monoallelic, are the end result of the same process, or it could be that they are the end result of two different processes. Even that is not known. Many question marks remain. Then I alluded to that in my answer to Paul's question. The coding region consists of one exon. So most genes, mammalian genes, mouse and human genes, are very complex genes actually to study and to work with because the coding region, the piece that encodes amino acids, has many exons, coding exons, and many introns, and they are put together by splicing. It makes genetic manipulation often very, very complicated. And the type of genetic manipulation we can do is greatly facilitated by the fact that there are no introns in the coding region. Now, what may that mean evolutionary? Well, they have been able to evolve very quickly, properly, and expand. Perhaps they went to a mRNA cDNA intermediate. We call that retrotransposition. So retrovirus then made the mRNA back into cDNA that integrated in the genome. Or perhaps by unequal crossing over, mistakes if you want, during meiosis, created new genes that then could evolve. So many unusual properties, okay? A lot of genes, just think about 1,000, in human it's about 400, still a lot. only one of the two alleles is expressed in a given cell, and that's, I think, quite well established, and no introns in the coding region. The wiring problem, we'll also discuss that. Also not understood, this is extremely diagrammatic. On the left are axons, so of olfactory neurons, forming the olfactory nerve, quote unquote. The olfactory nerve is not one single cable that goes from the nose to the brain, it's many little cables. This nerve consists of 1,200 different populations, right? Of neurons expressing each one of these 1,200 genes. And then at the other end of the nerve, they come out color-coded, completely sorted according to the receptor they express. And that I will develop in the next few slides. So a bit more scientific here. This is what I want to explain now. On the left side is the epithelium. In your nose, it is very high up between the two eyes, about the size of a thumbnail in human. It's an epithelium in direct contact with the outer world. Of course, it has to be, right? Odorants come in, diffuse through the mucus, and interact with these neurons. And then each neuron has, I'm going to draw on one here, a cell body. It has a dendrite, and here is the contact with the odorants, the chemicals. And on the other side is one axon that's not myelinated. It goes through little holes in the cribriform plate in the skull called the lamina propria. What am I saying now? Called the cribriform plate. And then in the brain, in the olfactory bulb, this axon will then enter one ball like that called the glomerulus. And if we follow another one, I will draw one in green here, dendrite cell body axon going to the cribriform plate, and it ends up in a different glomerulus. And one neuron has only one axon entering one glomerulus, which is also somewhat of a dogma. How many of these glomeruli are there? The latest count in adult mice is 3,600. We have 1,200 genes, so that's about a 3-to-1 ratio, which sort of makes sense, as you'll see in a moment. The diameter of a glomerulus is, in mouse, about 55 micron. Interestingly, in larger animals, you don't necessarily have larger glomeruli, you have more of them. There seems to be a finite size or an optimal size for a glomerulus. A glomerulus has no cells inside, it's an acellular structure. And in a glomerulus, you have synapses, hundreds of thousands of synapses. These incoming axon will terminally arborize, and then it will then synapse with the dendrite of the next cell, the second order neuron, which is called a mitral cell, or a tufted cell. It has a triangular cell body. And then the axon of this mitral cells will project to many parts of the brain, including the olfactory cortex. Oops. So it's a very simple system, this sensory system. You have a sensory neuron in the periphery, in the epithelium, that detects the stimulus directly. It then transduces into action potentials, electrical impulses, which travel directly into the brain and get processed in the bulb. And from there on comes the cortex, and then the mouse realizes, okay, this is the cheese, or this type of cheese. Okay, very, very simple, essentially monosynaptic pathway. How many of these mitral cells are there? They have also been counted, about 33,000. They're born in an embryo, mouse embryo, and they don't get replaced, so they're born just once. And if they die, they're gone. So for 3,600 glomeruli, that means on average only 9 mitral cells innervate one glomerulus. So you have a tremendous degree of convergence, right? You have hundreds of axons coming together in a glomerulus, and on the other side you have just 9 mitral cells innervating this glomerulus. Why did I write 1800? Actually, I have to update the slide. This was counted in the 80s by a French group. It was recounted 2 years ago by Charlie Greer with more modern methods, and they actually came up with double the number. Apparently the smaller glomeruli were missed. In humans, Charlie Greer at Yale also counted them recently. And they range in thousands of glomeruli, but there's a huge difference from human to human. So the jury is still out really on what is the average and the mean and so on in humans.
That also raises the question whether the notion of a glomerulus as a well-defined and delineated structure is actually reasonable.
You can in the mouse, sure.
Yeah.
But in '88, remember how you had to do things then, right? There was no Photoshop and so on, and they took every 10th section and drew it with hand and so on, and now you can do really three-dimensional reconstructions. There are some few difficult ones with glomela that look like a haltear, right? Just with a stalk and then two balls hanging onto it. Is that one or two glomeli? But I think by and large, they're discrete structures with stuff in between, empty stuff in between them. Okay, so you can more or less get a feeling for the complexity of the wiring problem. 1,200 populations. In fact, if we really are sticklers, it should be 2,400, right? Because each allele is expressed independently. And then here you have then these 3,600 glomeli, and that whole map has to develop quickly, because around birth already the mouse has a fairly decent sense of smell. It's important for its survival. Genomic organization, also very unusual, very weird. There are other gene families, the global and T-cell receptor genes, Mombaerts genes, and they're typically in one or a few clusters in the genome. They're called loci. Locus is the Latin word for site, for space. For autoimmune receptor genes, they're really all over the place. It's almost like parasitic DNA, and for mouse there are about 40 or more loci. Here are the different chromosomes, and then the bars indicate the number of genes per cluster. Some are all by themselves in the genome. These are very interesting ones, I think. Sort of genes lost in the middle of nowhere, but they're completely normal autoimmune receptor genes. There are a handful of those. These are the exceptions. Most come in small clusters. Some come in very large cluster. And two of the largest clusters here on chromosome 2. That is about 300 genes, all next to each other. And then this one here on chromosome 7. I think that's about 120.
And how much variability do you have there within species and between species?
The position?
Yeah.
No, that is conserved, yeah.
Highly conserved.
Yes. But if you compare this mouse genome, this is the mouse, compared to the rat, some of these clusters are in what's called synthetic regions, right? The region of this mouse chromosome that we think corresponds to that chromosome in rat, but others are not. You get a very complex map with a lot of arrows, and not sure really you can say this, this mouse and this rat, this corresponds to that. It falls apart already between these two closely related species. Now, the genes are located in any transcriptionally in any of the possible directions. So transcription can be like this. We call that head to head, or that could be tail to tail, or that could be like this. I think, is there another possibility? Yeah, and then this, right? So you have, no, I already had that one. So 3 possibilities, every possible transcriptional orientation. And the space between two genes is very small, just a few kilobase. And typically, although there is always, of course, exceptions in biology, there are no other genes in between. These are regions of the genome that are dedicated just for autoimmune receptor genes. Okay, now, the reason why I show that and dwell a bit on it is that it doesn't give you merely clues about how gene regulation may work. Come back to the other examples of gene families, like immunoglobulin T-cell receptor genes. They're all together in one big cluster. You can think about local mechanisms, local regulatory DNA sequences that control the expression of that cluster. But here you have so many clusters. In fact, a handful of genes are a cluster by themselves. They are the so-called singular genes. So this scattered organization doesn't give us clues immediately how it may work. It could be that one cluster on one allele, let's say this one in a given cell, is activated and is available for expression, and then the cell chooses one gene in that cluster, or alternatively, these 40 loci don't mean anything, and the cell, just by a magical mechanism, chooses this gene on this cluster directly. Okay, I hope you get a bit of a sense of what I'm after. Is it hierarchical? First the locus, the cluster, and then the gene, or does it go directly to pick one allele of one gene for expression forever?
But there might be another constraint here, Peter, that you haven't mentioned, right? Because some of the products of these encodings might be used also somewhere else in the body in the context of other functions, and that might then dictate some grouping along the genome.
Yes, there's more and more cases now, our field has been ignoring this for a while, of so-called autoimmune receptor genes expressed elsewhere. Now, if they're also expressed in the nose, then they're probably still autoimmune receptors. But if they're not expressed in the nose and only elsewhere, then they're for sure not odor receptors.
But it can be mixed, right? That, for instance, might appear in the kidney.
So there are about 9 of them, 9 or 10. It's a very interesting emerging story. One of them, a very interesting gene which pops up over and over again in many studies, MOR18-2. We have several knock-in and knockout mice for them, just by coincidence. We have a ligand for it. We really think it's an odor receptor. It's also expressed in the kidney in very specialized cells. And since we had a knockout, this person at Yale looked at this knockout and found a defective regulation of blood pressure. Now, who would ever have thought about that, right? Of looking at a knockout mouse with in a gene, one of these 1,200 genes, and measure blood pressure regulation. So there are more interesting stories to be developed there. But it's been largely ignored by our field, this extra olfactory expression.
Right.
Yes, and indeed, how would the regulation work there? Is it monoallelic there? We don't even know that. Is it monogenic? We also don't know. These are just a handful of papers in the literature thus far. The mice that we make, that's what we do for a living, so to say, make many strains of gene-targeted mice. Just show that up, show that very briefly. For those of you who don't know about it, it's a fairly long procedure. Was established about 25 years ago, Nobel Prize 2007, and basically the methods haven't changed all that much. You have these embryonic stem cell lines, which grow forever in culture. You can introduce DNA with an electrical shock electroporation. You can modify DNA, then identify the cells that have the mutation now in the chromosome on the desired spot, homologously. And then once you have these cells, you inject them into little embryos, what's called blastocysts. We get what's called chimeric offspring. They're chimeric because they have 2 different coat colors, typically brown and black. And then if we are lucky, it's a bit of Russian roulette, we get the mutation through the germline, as we call it. Which means, cut a long story short, the mutation we made by recombinant DNA techniques, so in the lab, is now present in the genome of this mouse forever, and we have a gene-targeted strain. There's very precise modifications. You can make changes in a single nucleotide in the genome using this method. The whole procedure from top left to bottom right takes about a year and a half. It's a lot of time is just waiting. Still hasn't gotten better over the years. All those new technologies now, as you maybe know about, TAL and zinc finger nucleases and CRISPRs, and those may accelerate things quite a bit. So one example here of a gene-targeted mouse strain, OMP-tau-LAC-Z. OMP stands for olfactory marker protein, and that's what we know about it. It's a protein that marks olfactory neurons, discovered in 1972 by Frank Margolis, and after 40 years, we still don't know what it does. It really shows hunting for a gene, function of a gene can be a very frustrating job. It's expressed at extremely high levels, only in mature olfactory neurons, very small cytosolic protein, and it's just a fantastic marker. This is what people use in the field when they want to stain histologically a section for olfactory neurons. So in that locus, on chromosome 7, I dropped in a reporter, as we call it, so a protein that reports the presence in cells. LAC-Z, you probably know about, beta-galactosidase. When you expose it to Xcal, a colorless chemical, you get a blue precipitate. Some of you may have done that in bacterium, right? These blue colonies is exactly the same. The bacterial beta-galactosidase converting Xcal into blue precipitate. You can also do this in tissues, and in this case, in a whole mouth. So this is literally a head of a mouse that we dip in this solution, a dissected head. The tau is fused to the LAC-Z is fused to tau. Tau has a very bad reputation because it's involved in Alzheimer's disease. If it's too much phosphorylated, it makes these tangles and it interferes with the normal function and anatomy of neurons. But that's what we clearly don't want to do here. Okay, we don't want to cause pathology in the cells. And in fact, after more than 20 years and looking at probably 50,000 mice in our lab, we have no sign of any kind of pathology. Why are we using then tau? It binds to microtubules. And microtubules, part of the cytoskeleton, are present in the axon all the way to the end. If you express beta-galactosidase in neurons, it goes in the cell body, a little bit in the axon, and then it stops. It just doesn't diffuse far enough, so you cannot see the whole axon. So the beta-galactosidase, the LAC-Z, has to be fused to something, at the level of the protein, in this case tau, to then make it what we call decorate the whole axon. Okay? So this is an axonal marker. Now what are you looking at now? Half of a head of a mouse, it's been cut off here and then cut in the middle. You're looking at the right head of the mouse that is then put like that on a dish. It's been stained with this X-cal. It gives you a blue precipitate wherever there is beta-galactosidase enzymatic activity. This is the main olfactory epithelium, which contains a few million of these olfactory neurons, each expressing one allele of one odor receptor gene. This is the cribriform plate of the skull. It's perforated. It has little holes, about 20 or so, through which go these little nerve bundles called phyla olfactaria. So it's not a single nerve, the olfactory nerve, but many little bundles. And then in red is the olfactory bulb, which is very large in the mouse. In human, it's a little bit larger, absolutely, but relatively compared to the rest of the human brain, of course, very, very small. The glomeruli in the olfactory bulb are not visible because the axons in this particular view are covering these glomeruli. So this is the OE, often called main OE, main olfactory epithelium. Okay, so the projections are from here to here, each neuron expressing one receptor, one allele, one axon that goes to the bulb and innervates one glomerulus. Just on the side, there's an interesting structure called the septal organ. It has about 10,000 neurons, and half of them express one receptor, SO1, which is a rule breaker because it responds to just about anything in the sigma catalog. Very, very unusual receptor. Makes no sense, of course, that a receptor binds to everything, but that's what we find in others. So some people have thought about the septal organ as some kind of an alarm organ, that when the mouse is sleeping, it's an obligatory nasal breather, by the way. It has to breathe through its nose. We can breathe through our mouth, but the mouse cannot do it. When something comes in there, a smell, then maybe it wakes up and starts sniffing. That's sort of the story that people come up with.
But
and then
where does it exactly project to?
Yes, to two or three big fat glomeruli for this SO1, and then a few here, all in the ventral part of the bulb.
And they're not preferentially driving, let's say, the local neurons in the bulb or so?
Not known.
Okay.
No.
Because it might set the baseline, right? That would be very helpful, actually.
Yeah, for instance, yeah.
But if you are in an environment that is loaded with all sorts of molecules,
Yeah.
it at least tells you, look, this is roughly the baseline pressure.
Not a good way of looking at it, yeah. Or conversely, when there is suddenly a new smell or a smell in the middle of the day, the mice sleep during the day, then it starts sniffing, right?
Right.
Yeah. Very difficult to study that. I mean, these are sort of stories we can come up with, but how do you study that mechanistically, right? Then this structure here is the famous VNO. And actually two of them, they're two cigar-like structures lying at the bottom of the nose. And they are specialized in the detection of pheromones. Basically, it deals with sex and violence, I like to say. These are chemical molecules that regulate between mice or other social organisms their behavior, which is typically aggression or no aggression, and sexual and also maternal. Now, the receptors expressed in the VNO are not the odor receptors of Buck and Axel. There are other G protein coupled receptors, another 300 or so. We have the V1Rs and the V2Rs, all discovered in the late 90s. They have nothing in common with the odor receptors in terms of sequence. Very large families again. And in humans, interestingly, they're all degenerated into pseudogenes except for a handful, which could be possibly human pheromone receptors. So a very strong case of evolution in the sense of a whole gene family that disappears in higher primates. And there's also some models and theories about why that has happened. So the vombronasal organ has about 7 cables, fascicles, that run over the septum. So by the way, our nose and the one of the mouse has a bone in the middle, cartilage, and on both sides tissue, olfactory tissue, and that's what you're looking at here. And then there's also on the wall of the nose, you also find olfactory neurons. So this is the septum that's on both sides covered with olfactory epithelium, and then this nerve swings here over the medial side. You can sort of follow it, and it ends in the back part of the bulb, a very flat part called the accessory olfactory bulb. Interesting part about this, I won't talk about the AOB further, that's a whole different talk. Projections are not going to the cortex, olfactory cortex. but to specialized nuclei, hypothalamus and amygdala, where they may affect endocrine circuits and so on. Because they're not going to the cortex, some people perhaps naively argue that this is not conscious type of olfactory perception, right? It is in the cortex that's where conscious perception is realized. And then finally, a very small bundle of neurons, only about 500 on each side, about 1,000. They were discovered by Martin Grüneberg, a German who lived in London in the 70s, now called the Grüneberg ganglion. It was kind of ignored or forgotten for about 40 years, and in 2005, 5 groups, including we, all using the same strain of mice from our lab, rediscovered this organ, and now that's a subject of quite a bit of a niche field. And indeed, this Grüneberg ganglion seems to be involved in the detection of alarm pheromones, in the sense that mice warn other mice that there is danger by secreting a substance that may act specifically through this Grüneberg ganglion. That's the latest of that. So the nose of the mouse is full of chemosensory structures. It's a highly chemosensory organism. We are much more visual. And higher primates, again, they live at night. They're nocturnal animals. They live in burrows. There's maybe not a lot to see there, right? So they have to depend more on their sense of smell and touch to find out what's going on.
But this Grüneberg ganglion is also using a dedicated set of receptor neurons?
Of receptor genes, yes. There's a few photoreceptor genes and even a taste receptor gene, strangely enough, that are expressed in these cells. They're unusual cells. They don't have real cilia and so on. But if you remove this Grüneberg ganglion, then you have a very strong behavioral effect surgically.
Which is, what's the behavioral effect?
That freezing upon smelling an alarm pheromone is no longer the case. The animals just freeze, right? Because there's some danger around them. Marie-Christine Boyer in Lausanne has done most of the work on that, and she had a paper I think last year with a chemical actually that mediates that alarm signal. So we believe that goes through the Grüneberg ganglion. So more or less, but I'm really simplifying here. Main olfactory epithelium is the garden variety of odorants. Septal organ, also kind of an alarm in a general sense, or maybe baseline. The VNO doing sex and violence, and then the Grüneberg ganglion, perhaps more in neonatal animals, that there's some kind of alarm. That's really simplified. This is a top view of the bulb. So there are 2 bulbs in the mouse and in human, and the projections are ipsilateral. So projections in the visual system are across, right, through the chiasm. And in the olfactory system, that's not the case. The left part of the nose projects to the left bulb, and the right part to the right bulb. Later on, there is crossing of information, but not early on. So this is anterior, front of the mouse, posterior. This is medial. This is lateral. Here in yellow, that's a part of the eye. So the bulb of a mouse is about the same size as an eye, but then squeezed a bit. Then you have all these little glomeruli, right? About 3,600. Let's say an average diameter of 55 micron. And then finally, in red, this vomeronasal nerve, which terminates here in this diffuse structure. Glomeruli much less well defined. There you really have a problem counting them. In fact, no one has counted them yet. Many little dots, AOB. And the projections from that AOB do not go to the olfactory cortex, piriform cortex, but to other parts of the central nervous system. And that's all related to pheromones. Again, also simplifying. Okay, now in this mouse that I just showed you as an introduction, very big long introduction, all olfactory neurons are labeled with this tau-like Z, this axonal marker. Now how about the neurons expressing one receptor? For that I developed, this was my postdoc with Richard Axel 20 years ago, almost to the day actually. I started the first week of September 93 with him. Came up with that idea of using a bisystronic strategy. You may have not heard about it. It's very useful, and I'll go over that in a little bit of detail. I got a very small piece of DNA called an IRIS, internal ribosome entry site. It's about 500 nucleotides long, and it comes from a mouse virus called EMCV, encephalomyocarditis virus of mice. The poliovirus, human poliovirus, also has a very well-known IRIS. And what the IRIS allows is that ribosomes, in the small and the large subunit, enter inside the message, well, one specific ATG translation codon and start making a protein. So the normal mode of translation is that the ribosome attaches to the 5' end. There is a cap here. It then scans the message, and then when it finds an ATG in a correct context, which we call a COSAC consensus sequence, it starts to translate a protein. And then when it hits a stop codon, it falls off. So it always starts at the 5' end, and then it scans and it starts somewhere. So thanks to the IREs, you can start internally, as the name says, right? Internal ribosome entry site. So that means from one message, from one mRNA, with this wiggly line, you can make two different proteins, two different polypeptides. In this case, the G protein coupled receptor, that 7' transmembrane protein, crossing the membrane 7 times, and something else, for instance, GFP. Why is that useful? Well, this is the unsolved mechanism. How does an olfactory neuron choose one allele of one gene for expression? We don't need to know this with this strategy. All we do is by gene targeting in embryonic stem cells, as I showed you a few slides ago. Insert this iris GFP, so precisely after the coding region, and then when a neuron activates that allele, it will turn green, and we can see it. Okay, so with this strategy, the bicistronic strategy and gene targeting, you do not need to know anything about gene regulation at that locus. That's the big advantage. Now a few examples. I'll show them each time with this acronym. This is M50. They have a new nomenclature that no one can remember. This is the old nomenclature. A few thousand neurons, you only see maybe 100 of them. In this particular picture, it's a hole mount. The cells are coming at you or going away from you, so you see them as one green dot. This is another receptor called P2, one of the first ones to be genetically modified, and then MOR23 is another one for which there is a very well described ligand called Lyrol, a synthetic ligand. What you see is that within the olfactory epithelium on the turbinate, so on the walls of the lateral walls of the nasal cavity, these neurons are not all over the place, but in a band, which we call a zone. For the last 20 years, we've been thinking there are 4 zones. That concept is now kind of degrading, falling apart a bit. There could be many more of them, a few dozen maybe, at the most 1,200, right? Each gene could have its own zone. There are about 20 genes that are expressed in a little patch, just a little circular area. Regardless of how many zones there are, we don't know what it means. Okay? I could just tell you, we just don't know.
But the zones are all oriented in the same way, or they start to crisscross and become more, let's say...
With the newer studies which we are doing, unpublished, that also falls apart. Yes, you have zones crossing other zones, and again, if you look back at the technology available not 20 years ago, this was done 93, 94, right? Photoshop was just there. It was very difficult to do digital neuroanatomy, and now with more modern methods and 3D reconstructions, you can see much better in 3D how a zone looks like. But it's kind of a dogma in our field. Unfortunately, it's already in textbooks before you know it. 4 zones, okay? No, and regardless of that, we don't know what it means. It would have been nice if the more anterior zone, zone 1, would respond to short-chain fatty acids, for instance, and the dorsal zone to long-chain fatty acids or aromatic structures or whatever, but there is no correlation. There is also no correlation developmentally or genomically. It's not the case that all the genes in one cluster, one of these 40 clusters, are expressed in the same zone. It's an enigma that came out of these in situ hybridization studies. It's important for us to acknowledge it. Why? For the gene regulation issue. So here is the border of the olfactory epithelium. The rest is respiratory epithelium. And in this most anterior part here, which I will color in yellow, there is no expression of that gene MOR23. So it's not quite right to say that a neuron has the choice of 1,200 genes, because these genes in the yellow area never express MOR23. It's not available for expression. Right? So instead of 1,200, maybe they can only choose between 50 or 100 genes or something like that. Maybe only 10 in the end, if we really look at that very carefully. So that's why I think this zonal issue is important to take into consideration for models about gene regulation.
But within the zone, the probability to find a gene expressed is sort of constant with the position within the zone. So
that also hasn't been looked at by proper statistics. It's mostly said in the papers, in the reviews, that it's random or stochastic. Now you know better than anyone to say that something is stochastic, you have to prove it, but molecular biologist looks at that like this and says it's random, okay? But there could be a pattern, it could be a very complex pattern in 3 dimensions. Remember, they're all convoluted structures, right? In 3D, they're not flat structures. So we could have easily have missed that, a higher order structure. There is one set of papers of Jörg Strothmann and Heinz Breer in Stuttgart that see for certain genes, the ones in that patch, that you find like multiples of 20 micron or so, 20, 40, 60, 80, but not 30 or 50. So someone has to look at that, who has a proper skills and wants to see a difference, right? If you want to say there is no difference, it's all random and stochastic, then of course that's what you will see. Our field, I must say, until a few years ago was really very qualitative. Many statements were made like that without any formal analysis or any statistics for that matter. In fact, you find many papers in top journals without scale bars even in the images. So, but it's improving now. Everything has to be quantitative in the end, and any statement you make in the end has to have a number, right? Then you can do statistics. So here is another mouse in which odin receptor M71 has been tagged. I'm zooming out a little bit here. This is the border of the respiratory epithelium, and again this whole anterior part has no blue cells, right? The cells in that part of the epithelium are not able to express M71. It's not available to them. We've been counting these cells. It's about 3,000, 4,000 cells per mouse. With quite a small error bar, actually. There really seems to be a program that says this gene is in 3,250 cells plus or minus 100 or something like that. And this gene is in 5,250 cells plus or minus 200. Another interesting aspect is probability. The axons migrate all over the bulb and they all come together in this little ball, this little glomerulus. Now, there have been several models, not that many, but a few models, how these axons can all come together here. One set of models argued or proposed that there is some information in the bulb that says this is like GPS coordinates, if I can say it this way. This is up, this is down, this is medial, this is lateral, maybe gradients, and telling, this is the position where you have to make a glomerulus. And what we prefer is that it's a self-organizing system where these axons are migrating and meandering around the bulb, touching each other, fasciculating, which they clearly do, and at some point, perhaps when there is a critical number of these axons, they all stop and make a glomerulus. That's the model that we favor and try to work out. The bottom line is it's not known yet by which cellular molecular mechanisms these axons make this glomerulus so precisely, so quickly after birth, and so reproducibly. In fact, the positions are very reproducible. They're not exactly stereotyped. It's not that if you have the position on one side, you can exactly find the one on the other side. There's a bit of noise there. So this is the top side of the bulb, left bulb, right bulb. This is anterior, this is posterior. This is the glomerulus you were just looking at, the medial one, and then you have the lateral one, and then here again a medial one, and here again a lateral one. Okay? And in fact, there is a plane of symmetry in the bulb. It's not a nice sagittal plane. It's twisted in 2 dimensions. So if we know the position of a glomerulus for a receptor here, then we can predict that the one on the lateral side of the bulb will be a little bit more anterior and a bit more dorsal, because this plane of symmetry is shifted. It's also not clear why the bulb really consists of 2 hemibulbs. That's not the case in zebrafish, for instance, another popular model for olfaction, or in Drosophila or honeybees and so on. But in mouse and in rat, there seems to be there is a clear internal symmetry within the bulb, and each receptor gene is represented in quadruplicate by glomeruli. Occasionally you find an extra glomerulus, never right next to it, but at a little bit of a distance. So I would say, depending on the studies and so on, and indeed how you define a glomerulus, this is anywhere between 2 and 3 glomeruli per bulb. And if we then multiply that times 1,200, provided they're all expressed in the nose, we come more or less to these 3,600 glomeruli. We're not quite there yet, though. There seems to be too many glomeruli. This will be sorted out in the next few years. Okay? So this is a fantastic example of biological specificity, right? What we think self-organizing map of these 3,600 glomeruli, depending on the receptor genes. The receptor is critical in there. We have made a lot of mutations in them, replace it with another one, made point mutations, all too much to go over here today. And clearly the receptor itself is critical in determining where this glomerulus is positioned. And that makes sense.
But can you say anything about the ordering of these glomeruli, or you will say something about that with respect, for instance, to the chain length of the molecules?
No. Yeah, that's the whole issue of chemotopy, right? Is there some kind of a logic in the bulb, and you find papers that say yes and other ones that say no, and there is a series of studies where you do with aldehydes, you increase the carbon chain length, and then the glomeruli go more and more posterior, like that on the dorsal side. But there is also examples against that. And
so you don't buy that?
Yeah, it's just too many that say one way and other ones that say another way. It's not clear. It depends also on the level you look at. For instance, if you look at let's say this glomerulus M71, and you look at the specificities of the neighboring glomeruli, they don't really. There's no direct logic there, right?
But across individuals, it's fairly invariant.
This map, yes. Yeah. It's not stereotyped. We have done a bit of analysis on that quite a while ago, and the positions of, let's say, this glomerulus would be within 20 or 30 glomeruli. So if you look at it over 3,600, that's less than 1%, which is not bad, right? But it's still not stereotyped, and we can really nicely show that, by the way, by crossing two mice with two different receptors labeled, let's say this one with GFP, let's say this would be receptor A, and then on the other side here, you would expect it also to be here, but sometimes it's on the other side, right? That's how you can show that the best, by looking at the relative positions of two glomeruli at a time, and then you see there's a bit of noise there.
Right.
That's why I don't like to use the word stereotyped, which means there's a copy, made on the other side. It's probably nothing like that in biology anyway. This is with GFP, and with GFP, particularly here in these glomeruli, we can do very nice reconstructions using a two-photon microscope. The confocal cannot penetrate deep enough. So this is an oval glomerulus. The long axis is maybe about 70, 80 micron. The small axis is about 30 micron. There is a hole in there. You can see that sometimes. Probably a blood vessel goes through it. And one sees that sometimes an axon bundle going to another glomerulus, through glomerulus. You have a few big bundles and a few small bundles. The smallest ones could be just one axon. It's always difficult to tell. They're only about 100 to 200 nanometer thick. And at this level of microscopy, in this age, it's about 3 weeks, there is no axons going elsewhere. So the precision of this axonal convergence is really high. One thing I warn you a bit, and these pictures are somewhat deceiving, we don't know the success rate of this whole process, right? For each 100 axons that leave the epithelium towards the bulb, how many of them end up in the glomerulus and survive? That is not known, and there is no method established to show that. It could be that that fraction is quite low, that there is tremendous selection, very quick degeneration of axons that miss target, and so on, and all of this is possible, and all of this could have been missed. This is sort of the end product which we see in a 3-week-old mouse. The monoallelic expression, I said that from the beginning, very unusual property. You don't find it very often in mammalian genes. We can show that easily by labeling them in two different colors, GFP and RFP, red fluorescent protein from another marine species. Whole mount confocal microscope picture of the olfactory epithelium. You find green and red cells. And if you do sections, they're really green or red, but not yellow. And you can count, let's say, 10,000 cells, and you find 5,000 that are green and 5,000 that are red and none that are yellow. So there's this equal strength, if you wish, of the probability of expression of both alleles. Now, for the smart ones under you, that means there are actually two populations of neurons expressing that gene. We're all working with inbred mice, or mice crosses between two inbred strains. That's artificial. A real mouse is, of course, not inbred. We do know there are polymorphisms in these receptors at the amino acid level. So in a given random mouse you catch in the field, it's very possible that M72 in that mouse will have two different versions of it, right? Two different polymorphic variants in the genome. And in that case, it may matter to separate their expression, right? Because then you would increase the signal-to-noise ratio by really making sure there is only one receptor type expressed per neuron. If they would be both co-expressed and they would be both slightly different, then they would have additive compound signal transducing properties. That's sort of the rationale that we give to this monoallelic expression. It improves signal-to-noise ratio. Difficult to show that, however. Okay, so the wiring diagram, it's getting clear to you now. Left epithelium, a few million neurons. Number is not known, by the way, in mouse or rat. 1,000 to the population, to be strict 2,400. One allele of one gene expressed per neuron. Each neuron has one axon, innervating one glomerulus. 3,600 glomeruli, 2 or 3 glomeruli per bulb. And then a glomerulus is innervated by about 9 mitral cells, second-order neurons that project themselves to different parts of the brain. So fantastic wiring problem. Not that much progress over the last few years. There have been some false leads also. That makes it worse, of course, in science if some people go in the wrong direction. And this is where all the sorting happens. They didn't really say that. Could have been that they're all sorted at the left side, at the level of the epithelium, that all the neurons expressing the yellow receptor are together and the green one, but that's not the case. It's only very close to the glomerulus that you suddenly find this sorting and axons of neurons expressing the same receptor come together. We can show this by electron microscopy. The receptor protein is expressed on the axons all the way to the glomerulus, so we do think that the receptor protein or receptor protein itself is intimately involved in this axonal wiring. But the precise molecular and cellular mechanisms are not known.
But now, Peter, now you give a very static description of sorting, right? It's
Yep.
more like, well, it's unsorted on one end and then sorted at the other end. But it's a dynamic process because these things have to grow in there, right, with time. And then they have to sort themselves out as they move through this olfactory bulb and try to find a glomerulus. So is that process itself already highly guided, or is that more like a random exploration?
Well, to do that, one would have to do time-lapse studies, and
Exactly.
no one has been able to do that technically.
Why not?
Well, I mean, if we would know how to do it, maybe then you could say why it doesn't work. But
Okay.
I mean, you have to have this whole structure properly, and good people have tried to do that, to take the whole tissue out, or the whole mouse, or the whole head, and
But if you have to guess, would you guess that it's also that process of growth of the receptor of the axon of the receptor neuron that's already highly guided? So the sorting is already done. There are guiding molecules. It's all sorted.
No, I don't think so.
Okay.
Because looking at many pictures in developmental stages, like postnatal, this glomerulus M71 develops between day 2 and day 3 postnatally, and you find a lot of axons going everywhere. Kind of a spaghetti-like structure.
Mm-hmm.
But we would love to see that, of course, in real time. You know, an axons making a bumping and then going back and all of that. But again, this is perhaps doable. Find a genetic way to look at the success rate. For every 100 axons that leave the epithelium towards the bulb, how many of them in the end innervate the glomerulus?
Mm-hmm.
And this could be very low, and then you can have a lot of selection, and no one thinks about it in those terms.
Right, exactly.
Everyone thinks this is a 100% success rate, one neuron, one axon wires, axon guidance, axon wiring, it's all perfect.
Mm-hmm.
And if it's only 10%, then you have a lot of mistakes made, a lot of randomness.
Right, exactly.
And there is no penalty to that. The epithelium has a lot of cell death going on throughout life, even in an adult mouse. When you look at different markers of cell death, of apoptosis, there's a lot of cell death going on, and there's a lot of cells wasted, but that's fine, right? Every epithelium is in constant regeneration. Think about this as the skin, right? Your skin, or at least of a mouse, is totally renewed within 14 days. That's been established. It's probably not as dramatic, but these neurons are being born and die all the time. It's also not clear, by the way, if the mechanisms of the establishment of the map, so late prenatal and early postnatal, if these mechanisms are the same as the maintenance of the map. Once the map is there with the glomeruli and new neurons are being born, let's say in a 6-month-old mouse, do they employ the same mechanisms to get to their glomerulus, or is it a different story? Do they follow a scaffold, and so on and so on? That's all. More research is needed, as always. The more you know, the more you realize how little you know, right?
Right.
Now we're going to deal a little bit with gene regulation. I'll just give you a brief flavor of it. I'm not going to go into many details, and you will get the take-home message very, very quickly. I haven't shown you yet the structure of an organ receptor gene. I told you there's no coding region, no introns in the coding region. It does have introns in the non-coding region. So there's part of the message that is not being translated into protein, and that's called non-coding. So this is the first intron, this gene, and this is the second intron. Okay? This is the structure of the gene. To find out what are the minimal DNA segments required for expression, we make conventional transgenic mice. So we take out this part from the genome, we clone it, this 9 kilobase fragment, we label it with iris star like Z because we have to be able to see and follow the axons, and we ask, is that block of DNA, does it have everything to control the expression of that gene? And that we do by conventional transgenic mice, which means you microinject a piece of DNA in an oocyte, in an one cell stage embryo. So there's no gene targeting, there's no precise integration, you have random integration in the genome. On Pietalaxi mouse I showed you at the beginning of my talk. This is the gene-targeted mouse for MO23, which you haven't seen yet. It makes a glomerulus here in the middle of the bulb. And then this is one transgenic line, TG is the abbreviation for transgenic, and this is the other one, made through ES cells, by the way. So yes, this 9 kilobase piece of DNA seems to have the information to dictate expression. And actually, we're asking a lot from that piece of DNA. First, it has to be expressed in olfactory neurons, not elsewhere in the body. Second, it has to be expressed in the correct zone, right? This dorsal zone of the olfactory epithelium. Third, it has to be expressed in a small number of cells, not everywhere in the zone, not in 5 cells, but somewhere in a few thousand cells. And then third, and then, is it third, fourth or fifth, the most stringent criterion, this glomerulus, the endogenous, the normal glomerulus, this one has to be innervated by the transgenic axons. Okay? That is by, when all these criteria are met, when you get expression only in olfactory neurons, only in the right part of the epithelium, only in a few thousand cells, we call that punctate, and when the axons innervate the correct glomerulus, then we say we have mimicked the gene expression, normal gene expression. That's been done only twice so far in the literature. Twice by us. No one else has been able to show all these criteria. In fact, the last criterion of these glomeruli has been missed in a few cases, but I won't go over that. So the 9 kilobase is sufficient, a little bit surprising. In a way, actually, we failed to get correct expression at the single cell level. This is slightly philosophical here. So the endogenous gene is labeled with green and the trend gene in red, and they're different cells. We can never get the trend gene co-expressed in the same cell. It would be yellow then. So it looks like the trend gene is just another odor receptor gene added to the genome, and when the machinery that is so fantastic at picking one allele of one gene for expression, when it picks that trend gene, it doesn't pick the endogenous gene for expression and the other way around, right? It's mutually exclusive. We kind of played evolution here. We added another gene, although it's the same one, to the genome. And this is the problem that other people in the field have too. How on earth can you now claim that your trend gene mimics the expression pattern, right? When it's never in the same cells. Well, here we go again, because the axons co-converge to the same glomerulus. The axons of the neurons expressing the endogenous gene, labeled green, and the ones expressing the trend gene, they project to the same glomerulus. That's why we think we have mimicked the expression pattern faithfully.
But to understand, this glomerulus that you now have built is like inserted among the glomeruli that have been developed based on the standard genetic expression of this animal.
Well, this is the endogenous one, right? So that's the green, and that's the green, the normal glomerulus, and that's now co-innervated by the transgenic.
Co-innervated, okay.
Co-innervate, that's the key point. Okay. And that's exactly the problem with the other studies, where they tried to do that too, and the trend gene created a new glomerulus elsewhere in the bulb. And then I think something was missing in the information in the trend gene, right? Either the expression level was too high or too low, or too many cells, or in the wrong part of the epithelium, or too early or too late, whatever, but you haven't mimicked the pattern. Yeah. So you need a gene-targeted mouse for that, right, to compare to. That's our reference pattern, the trend gene we compared it to. So when molecular biologists find something that expresses properly, they want to do what's called a deletion series, tinker with that promoter until it no longer works. Some people call it promoter bashing. Right? Just work on it until it gives you a defective, no pattern or a new one. So this is the trend gene now, a bit redrawn, about 9 kilobase. Here this is a shorter one where we remove this intron. still expresses properly. Then we remove this intron, we leave the non-coding exons there. Still expresses, very small piece of DNA now. Now we remove this bit, also still expressed. And then finally we lopped off the head of the transgene and we get no longer expression. So this is the only one here in this series that is no longer expressed, suggesting that the promoter, boy, were we lucky in hindsight, right? It's at the very, very 5' end of the transgene. That's where this so-called promoter is, the piece of DNA in which a transcription machinery assembles and then starts transcribing the RNA. Very, very, very small, about 500 nucleotide. Half of it is a repetitive element, so the unique sequence is only about 250 nucleotides or so. We did the same type of studies with another gene, M71. In fact, these are the only two for which this has been shown in the literature. All these features of transgenic expression. To the left, the endogenous gene. That's our reference glomerulus then. And here the transgene, and it's also again 9 kilobase. There we also made a deletion series, and we find that the minimal promoter is even smaller, only 161 nucleotides, which is very, very small for mammalian systems. What do you find? So now we have two small promoters. Then you ask, well, what's in common of them? Do I find a motif? Do I find the DNA sequence that they share? And yes, we find always, and other people too, in other studies, DNA binding sites for OE transcription factors. That these are 4 basic helix loop helix proteins. And more interestingly, they have binding sites for homeodomain containing proteins, of which there are about 260 in the mouse, genes at least. And those are the ones involved in patterning of the body and could very well be involved in patterning the olfactory epithelium. So this is the road that we and others are taking, looking at the role of homeodomain proteins in this autoreceptor gene expression, in particular the spatial patterning in the epithelium. I was just this brief part. Just remember the regulatory information to get all these features of expression in the olfactory neurons in the correct part of the epithelium in a small number of cells and with the axons converging to the proper glomerulus. All these have been shown with MOR23 and M71. Now, gene rearrangements. So the one receptor, one cell ID notions smells very much of lymphocytes. So in B cells, they make one antibody, and T cells make one T cell receptor. If a T cell would make 2 T cell receptors, you could easily get into autoimmunity. In fact, that probably happens sometimes when a T cell is activated, let's say by a virus, the second T cell receptor now attacks an autoantigen and causes autoimmune disease. But by and large, lymphocytes, one antigen receptor, one antibody, or one T cell receptor per lymphocyte. And how do they do that? It's an intricate mechanism of gene regulation that involves irreversible genetic alterations. The genome changes irreversibly. There are pieces of DNA that get lost, other pieces that get inverted. So it's stable, right? If irreversibly a piece of DNA is lost, then that change is stable. And it has 2 purposes there, is to make sure you have only one antibody or T cell receptor per lymphocyte, and you can also make a lot of variants. That's another story. Millions or even hundreds of millions of antibodies can be produced from a relatively small number of gene segments. From the very first paper, the 91 Buck and Axel paper, it was clear that in the coding region, which has no introns, there are no sequence modifications. That we knew. But what remained a possibility from 91 to about 2004 is that there was an irreversible genetic change in these neurons that would control the expression. Some kind of a switch, a piece of DNA that would be deleted, inserted, or flipped around, that would then, literally like a switch, right, turning on a light, would then turn on that gene forever. And that would be also an interesting way of thinking about one allele of one gene. It could only happen once in a cell. Now, how do you show that? The lymphocyte story was shown by my graduate advisor, actually, PhD advisor Suma Tonegawa, got his Nobel Prize for that in 1987. He showed that with cell lines. You had tumor cell lines, B cells and later T cells. You can grow lots of them, and it's quite easy, or at least now it's easy, back then not, to show with traditional biochemical and molecular methods that that gene has been changed. Again, with olfactory neurons, we cannot grow them, okay? That's a big problem in neurobiology. Mature neurons with mature phenotype and interesting properties cannot be enticed to divide and make a cell line so that you have lots of cells with the same properties. We cannot do that. They're post-mitotic. Now, when Dolly was cloned in 1997, I started to think, let's clone a whole mouse now from one neuron. We cannot make a cell line, but perhaps we can take the nucleus of an olfactory neuron and get a whole mouse out of it. A bit of a crazy idea, of course, but it worked. It took about 7 years. So what we did is to clone a mouse using a nucleus of a neuron expressing M71. That's one of our favorite genes, using nuclear transfer. I was lucky to be associated for many years with the guy who invented the mouse nuclear transfer technology, Teru Wakayama. He did so in Honolulu, of all places, University of Hawaii. I made many business trips to Hawaii, particularly in the winter. And then he came to my lab, and then after he left, we did this in our lab. And I'm going to go over that very briefly now. Also technically a little bit interesting. So before Wakayama, all cloning of sheep and cows and so on, in a way, was not really nuclear transfer. What was done is to take a cell and slip it under the membrane, under the shell, the zona pellucida of the oocyte, and then fuse the two cells with an electrical shock with Sendai virus. So it's called nuclear transfer, but it's really cell fusion, and whatever is in that cell, the mitochondria and the nuclear DNA, whatever you, goes into the oocyte. But what Wakayama did, he developed a true nuclear transfer procedure. So the recipient is a metaphase 2 oocyte. It's an unfertilized oocyte. He removes the chromosomes. It's called then an empty egg. And then he brings in the nucleus of a donor cell. I have a few slides coming up at that. Into that nucleus, into that cell, and into the egg. So without cytoplasmic contamination. And then after a delay of a few hours, he activates the embryonic cell cycle. And then in a small percentage of cases, you get a live mouse out of it. That is cloning by nuclear transfer. So these are the slides that Teru gave me. The left is the holding pipette, the blunt pipette that holds the mouse oocyte, and this shell that you see around it is this zona pellucida, under which, in other cloning techniques, another cell was inserted. The right is a sharp pipette, a nucleation pipette, and here is the endogenous chromosomes of the metaphase 2 oocyte. Very difficult to see, actually. You really need a trained eye to figure out where it is. If you remove not enough of that, then there is pieces of DNA left, chromosomes, and you get an aneuploid embryo, and it's not going to develop. And if you take too much out of the oocyte, of course, it won't grow either. So that's the whole trick, actually, is to do this enucleation properly. Aspirate, take out all the genomic DNA, and these cells survive, eggs survive for a few hours. Then you go to the mouse donor cell nuclei. These are cumulus cells, typically used for nuclear transfer experiment. We used olfactory neurons. And the key point is here to use a pipette that's too small. Very simple idea, actually. And by sucking up this cell, the plasma membrane ruptures, but the cell membrane, the nuclear membrane is much stronger, and the nucleus becomes a little sausage, right? So we can really squeeze into the pipette. Amazingly enough, that works, and there is not too much damage, and you can still get live mice out of that. So you can go back and forth, blow it out, take it up again, blow it out, and so on. Basically, you have a mechanical way of isolating single nuclei. So you suck up a few of these nuclei, about 7 or so, but of course only one will be injected per enucleated egg, then go back, and then inject with a special machine, a piezoelectric device. And here is this injected nucleus with the membrane around it. Again, more plastic than the membrane of the cell. And then, after a few hours, you have to activate the cell cycle. That's done with very non-specific methods, including 7% ethanol, or strontium chloride, 10 micromolar, or electrical shock. And then these neurons, these eggs will start divide. And in about 1 to 5% of cases, you get a live bone cloned animal out of it. It's a very low efficiency procedure, and no one really knows, after all these years, why it is so inefficient. So this is the paper that we published in Nature. This is quite a while ago now. And Rudy Jenisch and Richard Axel had another article in Nature, and we both basically published negative results. The title doesn't really suggest that it's negative result, but we meant to say is there are no changes in the genome of mice cloned from olfactory nuclei. Right? That's a very strong statement to say this is not happening, this is normal. We went through a lot of controls. It's something I don't recommend anyone ever going, to show there is no difference or something is not true, because you never know in the end, if you have looked at all the caveats and possibilities. So unlike immunoglobulin T-cell receptor genes and lymphocyte, there are no irreversible changes. And so this took about 13 years, and after that, this whole idea of gene rearrangements has come to rest, and now the focus is more on epigenetic mechanisms. So since this was a negative result, we needed to really build in a lot of controls. We employed another technical trick using genetic marking with the Cre recombinase. You may have heard about it. The Cre recombinase has recognition sites that are very short, so-called LOXP sites, and when it sees two of them in the proper orientation, it removes the DNA in between and stitches them together. So when we cross these two mice together, the M71 Irish Cre mouse, which we made, and this transgenic mouse, which we bought, then the cells that express Cre will delete this piece of DNA irreversibly, and now they express GFP from the chicken beta-actin promoter, which is a ubiquitous promoter. It's on all the time in all the cells, no matter what. So now the cells are green forever. That's called, in developmental biology, fate mapping or permanent genetic marking. Okay, so you can see where we're going to. At that point, that nucleus will instruct the expression of GFP, no matter what we do with it, and we have retroactively proof that we used the nucleus of a cell that was green. And that's particularly important when we get a normal mouse out of it. So the green cells in this mouse, this cross between the knock-in and the transgene, express GFP or have expressed it at the time of observation, and then when we do nuclear transfer, the cells will continue to be green. We made embryonic stem cell lines. So the green olfactory neurons, only a few thousands per mouse. Did the nuclear transfer, made a little blastocyst, called a little mini-me, and then from that clone blastocyst, make an embryonic stem cell line. Everything is green, and then from then on, we're off to the races. So these are the cell lines, they're all green. Everything is always green, that's the beauty about it. Okay, it's permanently genetically marked. We can really show to the reviewers, to the readers, and to you that we really picked the nucleus of a green cell, because from then on everything is always green. And now we have overcome this problem I alluded to already twice. You cannot make cell lines of neurons, so we cannot clone a cell, but we can clone the nucleus of a cell in the form of an embryonic stem cell line. So now we can grow up tens of millions of cells. This is the M71 locus. We can then analyze it by traditional southern blot analysis, do some PCR and so on. This is the promoter which we had identified in transgenic mice. We know where to look at, and we didn't find any changes. Okay? So no genetic changes nearby and no gene conversion, in which case there would be a third copy in the genome, and that would be the one that's expressed. It's all negative. Very, very difficult, very to want to submit to a paper, right, to a journal, said, you look, I don't find this and I don't find that. So we needed a biological correlate to that. We took those embryonic stem cells and injected them back into embryos. With another trick, we injected them in tetraploid embryos, which have double the number of chromosomes, because these cells don't do very well, and the injected embryonic stem cells take over the whole embryo. And out of that, we get beautiful mouse like this one, Harvey. So Harvey consists of 99.9 or maybe 100% of cells with a nucleus that once upon a time belonged to an M71 expressing neuron, right? So this mouse is cloned. Every cell in the body, or almost all of them, have the nucleus of one original cell type. And then we kill Harvey, unfortunately, and we analyze him, and we show that it's a normal mouse. We do some in situ hybridization. The details don't matter here, and we show that the neurons in these cloned mice do not all express M71, right? So that choice is not irreversible. They're not necessarily expressing that gene, and they can express other organ receptor genes. So that was the refutation of this model that was around in our field between 1991 and 2004. Are there gene rearrangements in olfactory neurons that control organ receptor gene expression? And we all now think that's no longer the case. Now, final story here, another 10 minutes or so, right? And about the P element. The P element it's about gene regulation. On chromosome 7, for many years, just from a historical point of view, we thought it was the promoter of these P4 genes, but we have never been able to link that promoter and the P4 message together, so it's not a promoter. It's a piece of DNA that we mistakenly took for a promoter. But because it was so interesting in sequence, it looked very much like the P3 promoter, we decided a few years ago to make a knockout mouse, to remove this piece of DNA. So this is a very clean and simple mutation. There is no iris tau like Z, there is no CRE, no GFP involved. We just remove this 370 nucleotides from the genome. Very, very small bit of DNA in the hope that it has an effect on gene expression. And it does, otherwise I wouldn't go for that. Now, the way to analyze this mouse was a bit complicated, and that's what maybe makes it from a technical point of view interesting. There's a complex cluster here with 24 genes. Here is the P element, the 317 base pair, which we found again by accident. We thought it was a promoter of an odin receptor gene, but it's not. Then we have a space here of 670 kilobase, and then is another cluster which we call P telomeric of about 41 genes. So 65 genes in total that we have to analyze. We make that knockout, that's the easy part. The difficult part now is how do we quantify expression? Some of you know about qPCR, quantitative PCR. It's very labor intensive, and to do this on 65 genes in one sample, too much pipetting. So around that time, fortunately, I learned about a new technique called nanostring. Does anyone of you know about that? This is a very cool technique. We didn't develop it and so on. We're just very, very happy customers. It's a small company in Seattle that developed this amazing way using single molecule imaging to look at up to 800 targets per reaction. And with target, we mean a type of RNA transcript, a message. It's all done in one tube, multiplex that's called. So qPCR is typical singleplex, one gene at a time. The detection is digital and analysis also, so it's numbers come out, integer numbers. Surprisingly, no enzymes at all are involved in this technique, including no reverse transcription, and it's highly automated with robotics. The probe design we do together with the company. There's a capture probe, which is about 35 to 50 nucleotides long, and it has on the 5' a biotin, and then it has a reporter probe, also 35 to 50 nucleotides, which has the barcode. Now, what the heck is that? Well, it is proprietary. Don't really know exactly what it is. It's about 6,600 nucleotides of heteroduplex of DNA RNA, and the RNA is chemically modified with Alexa dyes. So you have a stretch of 11 nucleotides that's made to fluoresce yellow, and then green and blue and red. These are the 4 colors. If you look at all the combinations and so on, you can make approximately 800 different barcodes, fluorescent barcodes. And these barcodes are visible and distinguishable through a simple light microscope. That's the amazing thing about this technology. And then we throw in our RNA. They form these molecular complexes, and those are then purified, stuck to a slide, counted, and come out in numbers. Now, how does that work? So the buffer and the code set is the set of these 2 oligos, the one with the biotin and the one with the barcode. They come in a box on dry ice from Seattle. We mix it with our RNA sample, total RNA. There's no polyandata plus, for sure no cDNA. Hybridize overnight at 65. That's all quite straightforward. Little hands-on time. Then we put it into a robot, a prep station, that does the purification with magnetic beads and so on. And in the end, it puts the samples on a slide and does a very short electrophoresis of about 5 minutes to stretch the DNA. So out come basically histological slides, and then they go into a microscope. That doesn't look anymore like a microscope. It's converted now into a scanner, and then it scans simply the slides, and then an algorithm detects how many times this fluorescent barcode was detected and how many times that one. Right? And you get, at the end, a simple CSV Excel file with the numbers. This barcode was seen 355 times, and this barcode 423 times in that sample. No enzymes and no cDNA. That's a dirty little secret in most gene expression studies, including now the popular RNA-seq. You're not sequencing RNA, you are first converting the RNA into cDNA, and then often back into RNA, or you analyze the cDNA, so you're not analyzing the RNA directly. But in this method, there is no such intermediate step. The RNA itself is analyzed directly. So the mRNA here, that's what we provide. We prepare, purify, the capture and reporter probes, mix them together. Then these funny tripartite structures are then bound with the biotin to streptavidin-coated cover slips. So at that point, they're just immobilized in one dimension. No, they're just really, but they can still move in this dimension. And then comes another trick, another binding gets added, this little electrophoresis, and now you stretch the DNA. You can actually stretch DNA. If you stretch it too much, it breaks. Here they stretch to about 70%. When you do all the calculations and you ask 6,600 nucleotides of DNA, RNA duplex, stretched to about 70%, that comes to about 2 micron. Now 6 segments in there. In fact, these are old technology was with 6 different segments. So divided through 6 is about 360 nanometer, and that's perfectly within the limit of a light microscope, right? You don't need any special super resolution microscopy to distinguish a little segment of 360 micron in yellow from another one, 360 micron in green. Now, we are not looking obviously through the microscope and counting millions of barcodes. That's all done automated by this algorithm that's in the scanner. And it pops out again this CSV file with the counts. These are integer numbers. Then there is positive controls and negative controls and normalizations. A lot of stuff I spare you with to make sense out of these numbers. So this is the biggest code set we have made so far, which we call Gorilla. 600 genes, most of them OR genes, many of them in the cluster, 8 reference genes for normalization, and these probes are all against the coding sequence, which I'm very happy about. We are not using 5' and 3' non-translated region. It's another dirty little secret from any gene expression studies like microarrays. They go into the 5' and 3' non-translated sequences because they're more variable. But these are not well defined in many cases, often only computationally. And if your transcript just misses that piece of 5' or 3' non-translated region, then you're not seeing it. Okay? Whereas if your probe is against the coding region, for sure, you will detect it in all cases. So here is how this actually look like. The position of these fluorescent barcodes doesn't matter here, right? They're all just spread out and stick to the slide, occupy space. It's a little bit crowded. Half the amount of total RNA is better. This is much better. Now you have too much empty space, unused space. And if we don't add RNA, we still get little dots. These are the fiduciaries, the landmarks that are used to align those 4 different pictures. So it takes a picture with red, blue, green, and yellow, aligns them then. You have a pixel shift, right? And then to align that, you have these little beads. And then occasionally you find a probe that made it for some reason and got stuck and shouldn't be there. But the background is very low, often in the single or double digits. So 1 microgram for our studies in this code set seem to be optimal. So we're looking at mice that are 21 days old, not 20 or 22, just 21. They're all in inbred BLAC6 background, which was very important for our studies because the reference genome, the genome that is the best sequenced so far, is still always the BLAC6 mouse. And then again, very big code set with 600 genes. I'm just going to skip that control experiment in the interest of time. This is the main slide. I will just build that up over a few animations. On the x-axis is basically the amplitude. That's the signal, the number of counts in wild-type mice, WT, and we do a log2 average. That's typically done in gene expression studies. In this code set come these so-called alien sequences that the company adds in there. These are foreign sequences that don't exist in nature and that are used as positive controls for the purification and detection steps in 4-fold dilutions. And then on the y-axis, the vertical axis, we do the fold change, a typical nomenclature of gene expression studies. So in this case, we do mutant, the mouse lacking that 317 base pair piece of a wild type. If mutant over wild type is 1, if there is no change, then the log2 is 0, right? If mutant is less than wild type, so if it's less than 1, then we get a negative thing. So here is no change, and then anything going below is reduced in wild type compared to mutant. That's how you have to read this slide. Then we have the negative controls, also alien sequences, probes for target RNAs that should not be in the samples. And then you can say, for instance, I take the highest negative control, and that's considered my background. And I point out again how little that is. That is 2 to the 5, so 2, 4, 8, 16, 32. That's only 32 counts for the highest background that is seen in this particular experiments. And then we have all these hundreds of genes now. Mutant is the delta P, the mouse with the knockout of the 317 base pair, over wild type. The sort of old dance, we call it a cloud of gene expression values. We then do statistical analysis. It's too far to get into, but just one little point. Many people in the gene expression field define a cutoff. Let's say If it's 1.3-fold increased or decreased, I call it significant, biologically significant. We have modified some statistics from used for microarrays, where we ask the question, is the observed fold change statistically significantly different from this chosen 1.3-fold? Hope you understand the difference, right? So typically, microarrays is done fold changes of 1, 1.1, 1.2, 1.29 we ignore because there could be noise, but if it's 1.3 we believe it and everything above, right? But we ask, is the fold change we observe, is it significantly different from 1.3? And that's a different question. It is more reproducible. So by using this kind of statistics, moderated t-tests, we find 10 differentially expressed genes out of these 500 something. And to our great satisfaction, they're all close to that P element. We didn't know this in advance, okay? That was just blindly, totally openly analyzed. You can see that easily if you plot all the genes through the genome, on these 40 different loci, there is kind of a dip in gene expression on chromosome 7, and that's where that P element is located, and all the other genes are non-differentially expressed, mutant over wild type. Remember, there are two clusters around the P element. There's one with 24 and one with 41, so total was 65. And the P element is located right here. The wild type, these are the numbers in wild type mice. These are in mutant mice in red. And the ones in the telomere cluster were all unchanged. That's a very, very nice control for us. So the nearby cluster, only 670 kilobases away, none of these genes are significantly different between wild type and mutant. But in our cluster, so this is the location of the P element, we find 10 genes that are differentially expressed. 9 are down and 1 is up. And these are within approximately 200 kilobase region. Now there is another element known from other studies, which we also published, called the H element, for another cluster. And we found by these and other studies that it also has a range of about 200 kilobase. This could be complete coincidence, but we have two cases now. So let's make a quick calculation. We have 1,200 odin receptor genes. They're on average 25 kilobase apart. So that's 30 million bases in the genome dedicated to odin receptor genes. And if every 200 kilobase you find such a regulatory element, we're going to have about 150 of those little pieces of DNA all scattered through all these loci that somehow affect this gene regulation mechanism in a way that we don't understand yet. This is my final data slide, just on time. Makes an important point, I think. On the x-axis is now nanostring. Again, when the log2 is 0, there is no change, right? But over mutant over wild type. And negative means mutant is lower. You can also count the number of cells in the mutant mice expressing a given odin receptor gene by in situ hybridization. Now that's very laborious and very boring, particularly the counting part. You have to do every third or fifth section, label in green and in red, and then manually look in on the screen and say that's a cell and that's not a cell. And the available cell counting algorithms are not great. In fact, the human eye is still better, and if you ever have one, please do it, because to get the data on the y-axis is about 1 month of counting. Okay, by a person looking hours and hours per day and saying that's a cell and that's not a cell. And finally, the numbers were there. They were all, I cannot say really analyzed blindly, but analyzed without knowing what probe was used and what mouse. And then we did these fold changes for the cell counts and compared them to the RNA counts, and we found this amazing Pearson coefficient at close to 1. So to show this with this particular example, that's also called P4. And so the P element is between P3 and P4, that's why we call it P. It is down in this crude RNA mixture that we purify 2 to the minus 4, that is 16-fold down, right? 1 over 16. And if we count the number of cells in that mouse, it's also down 16-fold. That's all we want to show with this picture. But it's an important conclusion, we think, is that without the P element, the number of cells is reduced for 9 out of 10 cases. In one case, it's slightly increased. And the amount of RNA for that whole population of cells is decreased to the same factor, which means the average amount of RNA per cell is not changed. That's why we'd like to make this final proposal here. working hypothesis. The P element and that other element, the H element, regulates the probability of gene choice, which is a kind of a different concept to think about mechanistically. The number of genes, of cells that express a given gene from a cluster, but it's not regulating the level of transcription after the gene has been chosen. About 30 years ago, enhancers were discovered here in Europe by Chambon in Strasbourg and Schaffner in Zurich. You may have heard about it, enhancers. They are enhancing gene expression. Typically, one thinks about them as acting on a promoter and literally enhancing, increasing the expression from a given promoter. There is a small number of studies that have suggested that some enhancers can actually regulate the probability by which a given promoter is on or off in a cell population. And we think our elements here are great examples of those. They are not regulating the strength of a promoter, the transcriptional level, but in a binary way if that promoter is expressed or not expressed. Okay, I just leave you with that. That's a lot of stuff here on this slide. It has a number of predictions which we are testing, which all require some very tedious and long genetic manipulations. And this is one of the heroes in the story, Harvey, one of the cutest mice ever we could take a picture from. It took hours and hours to get that. Maybe we have time for a few questions. Thanks.
Very good. Thank you, Peter. All right, questions. So Peter, to start with the last part, so you spent an enormous amount of effort to get a handle on this P element, right? And the P element, in the end, would be sitting at the beginning of a chain of expression, and it sort of defines the fate of that particular cell, whether it's going to express this receptor or another one.
Don't think I exactly said that, but it defines the probability that in the epithelium there are, let's say, 6,000 cells expressing that gene, and without the P element it will be only 500 cells. Yeah.
But it's part of some selection chain, if you
Yes.
want, right? That sets the fate of the cell. But in itself, you would expect a controller of the P element as well. So you would expect some hierarchy of switches in which this P element sits somewhere, I would guess. So how do you think about this? Well, what's the hierarchical structure there? And what's in the end the key signal that has to sort of enter that cell to say, okay, we're going to do this?
So the one popular way of thinking about all this is that there is some factor in the nucleus that's of limiting amounts available in limiting amounts, right? In the end, maybe only one singularity, which is not easy actually to think about again molecularly, and that can only interact, let's say, with one autoreceptor promoter, and with the P element is somehow involved in that and controls that probability, but it's competing with other ones. Complicating all of this is that it depends on where the cell is located in the epithelium, right? Remember that? That's totally not understood, how that's regulated. So we like to think about gene regulation, or at least our lab, as indeed a hierarchy, many steps, many levels. There's no simple answer to this, no like, this is the way it works. And these elements are just one aspect to it, which control probability.
But then would you see that the chain of regulation as being very specific to the olfactory system, or a very generic kind of switching mechanism?
That's an interesting question. Yes, if it's only for autoreceptor genes, maybe it's not so profound interest. But many things in biology are used in other systems.
But it would look like complete overkill, right? If it would only be used in the olfactory system.
But as I said, the traditional way of looking at enhancers, if you look in a textbook, is it enhances the transcription of a given promoter. But virtually all these studies are done in bulk cell populations, in transfected cells in a dish, or in a transgenic mouse, where you express something in the liver and grind up the liver, but no one ever counts the cells in that population that express the gene, yes or no. It's an average, right? So that's why these enhancers, maybe these results draw again interest to these older ideas and data that enhancer could really be a switch that says this promoter is active or not in a cell within a population.
But then that choice is also irreversible.
Yep.
Right? So how do you realize that? Because in some sense you cannot revisit that same chain of decisions anymore. Then once it's set, it's set.
Yep.
So how do you do that?
There has to be probably epigenetic and methylation and histones and all of that and all these goodies that are one leads to the other. So indeed there is no way back anymore.
Okay.
in this decision-making chain, right?
Right.
If you wish.
It has to
Yeah.
be, right?
Yeah. Now there is one or two studies that suggest gene switching. So from Richard Axel's lab, but they were technically not done so well, these studies. It could be that in immature neurons or in early neurons that there is some kind of they still have a bit of the choice that express this gene or that gene. They haven't made up their mind, and that's called gene switching. You find a few papers about that.
But now, you don't only want to switch your receptor neuron, you also want to make sure that in the receptors they express, you maintain a certain level of variability. Right? You don't want to have all these receptors be identical, because the ligands you want to be sensitive to will show some variability. So you want to, now, the decision tree that you gave us with this P elements is sort of only switching the identity. But now within that, the expression of this whole control loop, or the control system, there must be another element that still says, oh, but within this we still want to have some variability in how we express these receptors. So do you see that as part of this chain in which
So you mean that all the cells expressing, let's say, the P4 gene, there's still some variability within them?
Yes.
Well, maybe there is a second receptor co-expressed with some of them. Right? Back to the old dogma.
Okay.
It could be at a different level. That is something that we really cannot exclude at all. If there is a second or multiple receptors co-expressed at lower levels, that would have been missed by any method, right?
Okay.
If you do single cell RT-PCR, you only get the one that's expressed the highest, that's also amplified completely non-linearly. That would be one example of it.
Right.
Completely heretical at this point in time, and would be a bit annoying for our current models of olfactory coding. Yeah, if a given OR gene would be co-expressed with other ones, and there would be variability within the population. Yeah.
So another thing, so to go back to the earlier part of the talk, so when we think about how this whole now olfactory bulb is being organized, and in some sense we focus very much on the wiring of this whole system. One argument is from an evolutionary perspective, one goes, well, this olfactory system is actually very old, right? It has emerged very early in evolution.
Yep.
And we could think about it as being some sort of, let's say, template along which you also can build other sensory systems. Do you look at it in those terms, or do you really see these more as sort of specialized systems that have not, that audition or vision has sort of piggybacked on organizational principles of olfaction?
In evolution, it's indeed probably one of the most primitive, if you wish, maybe also the most complex, because single cell organisms can have chemical sensing, right? They have to find a nutrient, like bacteria, and go towards it, or little nematodes. They either go towards the source or they go away from it because it's danger. I mean, all in life is basically, survive, don't be eaten, and reproduce, right? So, all these very simple requirements, olfaction is critical. Now, with regard, if it's a model for other sensory systems, it actually annoys me a little bit that there is, in our field, so many analogies always made to the visual system and also immunology. Many times it works, but many times it doesn't work, and I don't find it all that useful, actually, to constantly make these analogies with other systems, because often the analogies are not there, right? It's only occasionally when there is like activity-dependent formation of circuits. Yes, some aspects of it are maintained in sensory and in the visual system. And with regard to other organisms, glomeruli are also present in insects, right? They're very small there.
Right.
The whole olfactory antennal lobe of a Drosophila fly is as big as one glomerulus in the mouse. You have a huge difference in scale. But if you really look below that surface, this obvious similarity, it very quickly falls apart.
Right.
And also experimentally, to then say, let's do this experiment because of that, it doesn't give you, in both ways, not much inspiration. That's the way I like to look at other systems, right? Does it give me an idea to do an experiment? And if it doesn't, then maybe there is not much analogy there. It's very superficial, but it's always good for reviews and texts, and it's evolutionarily conserved. We have to write that, isn't it?
Yes.
So now looking at all this anatomy, but also and how this is expressed, and the gene expression, I mean, was very impressive. But now for me the question is, what does this mean from a computational point of view? Because at the end, the animal has to behave in a certain way. So my question is, can you, from the anatomy and from all this mechanism of gene expression, actually tell us something about how it helps the animal to survive? And what's the coding you would expect them from such anatomy? Or
yeah, it provides a template, a basis to think about these studies. Some of them have been overinterpreted as if there is a spatial map of glomerular activation, right? As if this particular pattern of glomeruli means this for a mouse or a rat. That we cannot conclude from all these studies. It just provides you a template. In general, I must say that olfaction seems to have an overkill of coding capacity. You have in the mouse 1,200 odorants. A given molecule stimulates easily 10 receptors, if not more. The scent of a flower, for instance, is hundreds of different molecules. So you have tremendous coding capacity, many combinations. And how is this poor mouse with its cortex deciding then what that smell means, right? And what to do with it? Maybe that's what you're getting at. And, or if, as I say it often to lay people, we don't understand at this point in time how the smell of a banana is encoded and why different bananas all smell to us like banana and not like a lemon. And that very simple question we cannot explain yet. These categories, right? A small number of discrete, meaningful categories that have to be extracted from this enormous amount of information that is coming into our nose. I'm glad to admit that.
Do you have other questions? Vicky, you had a question?
In insects, there are some certain glomeruli, for example, that are specific
Yeah.
for pheromone detection, for instance.
Yes.
Is there any set of specific glomeruli for active behaviors?
Well, the voronasal system, that's the VNO is specialized in that, and there are these about 300 receptors in the mouse, about 150 V1Rs, 150 V2Rs, and they project to the accessory olfactory bulb, which has thousands and thousands of small glomeruli. You cannot even count them really, but it's not like this fantastic specific glomeruli in the moth, for instance. That's probably what you're referring to.
Yes.
No, there's nothing like it that we know of in the mouse or the rat. Here again, we have an overwhelming number of receptors and ligands, and we can't quite figure out how the system figures out, what is meaningful and what to do with it. It seems a lot.
But it's fair enough to say that in some it's still an open question. Because as you showed us earlier, you still have quite some substructures within this whole olfactory system that are not so clearly understood. Right? So it might as well be that you have actually also highly dedicated labeled line pathways that function a similar way. We just at this stage
that's what I said. At this point we don't know that. Yeah, there could be some very very specific chemical molecules with very specific receptors and very specific glomeruli, which you then can manipulate experimentally.
Mm-hmm.
Yeah.
Right.
But again, I tend to see this there's just so much coding capacity, so much overkill, so much excess, and how, what does a mouse do with all of that was the question of the other person. What categories of information are retained from that?
Well, like for the mouse that you study, what do you know about the complexity of its chemical world? How many cues will it have to detect in there? Or is it dozens, hundreds?
And which are meaningful, ecologically?
Yeah. What's the common wisdom in your field among your colleagues?
What is the natural? There's a whole workshop actually starting today, I think, in Hanover, dedicated at the auto space, right? And,
right, exactly.
Yeah. Much of these studies are anthropomorphic, I must say. What we tend to use as stimuli are nicely smelling odorants to begin with, from the perfume industry, right? But those are not necessarily relevant for the mouse.
Right, exactly. Other questions? Okay, please.
Is it possible that the P element is a transposable element?
Yeah, very good point. Yeah, it could very well be. And in what sense are you thinking about that?
I'm thinking yes, because it seems like an ectopic factor coming in a genetic region and starting to regulate it. I think epigenetically with ways of methylation, and I think I understood that it's a spatially related.
Locally, yes.
The position of the P element is relevant for the way it regulates the genes.
There are some hints that go exactly this way. I have a student working on that, and it could be that these genes indeed expanded quickly throughout the genome with not transposon, in this case, retrotransposition, and or illegitimate crossing over and so on. But then you still have to regulate their expression, right? And that could be by very small pieces of DNA that hop around. And they are, it's not that, I forgot to mention that, yet you can, there's some motifs in the P element, again homeodomain by the way. It's not that you can just look at these motifs and find all the other 150, unfortunately. There could be very small pieces of DNA and very degenerate, but they could indeed be of transposable origin long time ago. To regulate each time within, in the vicinity, let's say 200 kilobase, the expression.
So to finish off the discussion, I'll finish it up. You have said now several times, also in the discussion we were having, that there is this amazing complexity to the system that almost seems ill-justified, right? So how do you look upon that? Is it like, okay, this complexity is there because the system is regulating aspects we just don't understand yet? Or do you see it also more in these terms like, well, it's a bit like what we were discussing with the previous talk, like, well, it's like this tinkering that's going on, basic elements are being reused and used in different ways, leading to maybe unnecessary complexity, but it works. So which side of that debate do you stand if you look at that complexity?
Yeah. Well, to turn the question around, I mean, the next few years or decades will be great when all these genomes are sequenced, and you can actually look at that in a different way, right?
Is that really the approach?
Or between different strains of mice, for instance.
But is it really the solution?
No, but it's one way of looking at it. And I can tell you, the person whom I think wrote a fantastic review about it, and all the men concluded that from his studies, it seems random. But
you mentioned.
Okay, so that means, irrespective of that, how can you rethink about evolution, pressure working on a family of 1,200 genes, including receptors with overlapping specificities, right? If this M71 gene is lost in a mouse, or has been added to it, will that mouse have a selective advantage over other ones? It's very hard to think about that, right? It seems so incremental also, and so meaningless. A drop in the bucket or something like that. Maybe that goes along with. But from a point of view of gene regulation, axonal wiring, so very fundamental molecular and cellular, we think this is a fantastic system to work on, irrespective of what it does actually. As you maybe noticed, my talk was not really about the sense of smell, right? We just use it as just a great system to study these properties.
Exactly right. Very good, Peter, thank you.